Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Ellen Capon, Editorial Assistant at Drug Target Review. Today's speakers will be Vanessa Porcalab, Biophysics Director at Eurofins Discovery, Alexis Moreno, Research Scientist at Eurofins CALIXAR, and Maud Sigalo, Research Scientist at Eurofins Discovery. Following their presentation, we will move on to a live question and answer session where you can pose questions to today's speakers. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So, without hesitation, may I pass over to our speakers.
Hi everyone, it's a pleasure to be here today. We will address in this webinar today a fragment-based screening project on challenging proteins that are GPCR. It was a collaborative work done by our expert in membrane protein production and biophysics. Before starting, a few words about Eurofins Discovery. We are a CRO offering a complete package in integrated drug discovery, from hit finding, including virtual screen, HTS, fragment-based screening, and follow-up such as chemistry, in vitro pharmacology, safety, ADME-PK, and up to custom assay with protein production and biophysics. Let's talk now what are the role of biophysics in drug discovery. The biophysics are commonly used in hit finding, hit to lead and lead optimization phases. We are offering custom services for small molecule projects, from standalone project to integrated drug discovery programs. So today we are going to present you how we address a fragment-based screening targeting membrane protein. Before starting, we will do a quick introduction about us. My name is Vanessa Porcalab. I'm a head of biophysics at Eurofins Discovery Rep. I have a background in biophysics, working from fragment to antibody characterization. For several years now, I'm driving biophysics projects and leading a team of scientists involved in integrated drug discovery programs. So it's a pleasure to be here today with Maud and Alexis. So I will let Maud introduce herself.
Thank you, Vanessa. I'm very happy to be speaking with you today. My name is Maud Sigalo. I'm Biophysics Study Director. I work closely with our client to define the most effective biophysical assay development strategy for their project. I have a PhD thesis specializing in the perception of sweet and umami tests mediated by GPCRs. And then I did a postdoc focusing my research on biochemical characterization of nanobodies binding to the cystic fibrosis transmembrane regulator for therapeutic purpose. Before joining Eurofins Discovery, I was project leader in a biophysics at a biotech company working on oncological drug discovery. And the last speaker today is Alexis. Could you please introduce yourself, Alexis?
Thank you, Maud, and thanks everyone for joining this webinar. I'm Alexis Moreno. I am a biochemist with expertise in membrane proteins, and I am working on custom and R&D projects as a research scientist at Eurofins CALIXAR for the development of therapeutic target proteins.
Thank you, Alexis. Before talking about our program, I would like to define with you what are the role and the scope of biophysics. It's commonly used in drug discovery, from compound profiling to mechanism of actions. Working mainly with recombinant proteins, the biophysics technologies allow us to characterize hits. Our goal is to understand the relationship between kinetic of binding, mode of action, and molecular interaction. Thanks to our complementary biophysics techniques, we can look on the strength of the binding, the kinetics, the thermodynamics, the stoichiometry, but also the protein quality. Based on the request and the aim of the project, those information as KD, melting temperature, K on, K off bring valuable data for compound ranking and selection. As example, on the next slide, so we are offering different projects. In the hit finding stage, we can perform a fragment-based screening campaign with a defined set of fragments that will be a starting point for fragment growing, or we could also introduce biophysics as an orthogonal assay after an HTS. In that case, after validation of hits using functional assay, we can confirm the hits with their binding properties with the targets. Then, regarding hit-to-lead and lead optimization, we can provide support for medchem analysis and insights into the mechanism of action. We know that kinetics have a significant impact in pharmacokinetic and mechanism of action. If you have a specific request, several approaches can be also investigated with the mechanism of action, and we can, for example, determine the hit selectivity for the target, the stoichiometry of the binding, or we can also take a look if the compound is an allosteric or orthosteric binder. So all of those projects and hit screening are possible thanks to our cutting-edge equipment. Here at Eurofins Discovery, we are working with the most recent biophysical technologies. From the left to the right, we could find the BioCore 8K+, the Dianthus for MST trick technology, including Spectral Shift, the PIC ITC, and finally the BioRad devices. All of those technologies are complementary and have some advantages and some disadvantages. Recombinant protein can be immobilized on the chip, whereas for MST, the protein is in solution, and for ITC, we will have also the protein in solution, and for TSA or DSF, we incorporate a dye that will monitor the unfolding process of the protein. So for screening and determination of KD value, BioCore 8K+ and Dianthus fit perfectly for the need with their 384 well plate format for compound testing. Additionally, we can also extract several types of information based on the technology. We can extract some binding constant, kinetics, K on, K off, thermodynamic parameters, stoichiometry, and melting temperature. So here for fragment-based screening project, SPR, MST spectral shift, and TSA can be used as primary screen. And for membrane protein, we recommend to use the MST spectral shift approach that is a detergent compatible system. And the main advantages here is the membrane protein is not immobilized and it requires a limited amount of recombinant membrane protein. And finally, prior to start such project, we used DSF and DLS to validate the protein quality and stability. So let's start. We will present you today an example of fragment-based screening using spectral shift for membrane proteins. And here we will focus our work on HOA adenosine receptor, that is a GPCR. So why and how to identify novel GPCR starting point? So that's a good question. Compared to a conventional HTS, fragment-based drug discovery offers an alternative strategy to identify leads based on fragment hits. The main advantage of this approach is the property of fragment libraries that offer better coverage of chemical space and give the chance to identify new chemical starting point. Here is depicted the HOA adenosine receptor fragment-based screening approach. And this target is a nice example of GPCR for which crystal structure were solved, and it brings an acceleration in the development of drugs to treat cancer, CNS, and cardiovascular diseases. So now we will review step by step our screening cascade, and we will start first by the protein production part, and I will let the floor now to Alexis.
Thank you, Vanessa. First, let me introduce Eurofins CALIXAR. Eurofins CALIXAR is specializing in the production of recombinant membrane proteins. To this end, we express our protein in the expression system of choice, be it primary cells, mammalian cells, insect cells, yeast, or bacteria. We offer our services for the custom production of soluble proteins as well as membrane proteins, for which we have developed proprietary detergents, polymers, and stabilizers to increase the solubilization rate and enhance the protein stability. These molecules allow the purification of membrane proteins in detergent micelles or in synthetic nanodiscs, such as SMORP, with the common goal of isolating full-length and wild-type proteins while maintaining their activity and stability without the need for engineering mutations or truncations that would alter the protein. We also offer the possibility to reconstitute the purified proteins into detergent-free systems, such as MSP lipid nanodiscs, proteoliposomes, and amphipods. This fully native process aims to provide stable and functional protein targets for small molecules and antibodies development, structural studies, bioassays, and vaccines. Here, we are going to show you how our recombinant proteins are used within Eurofins discovery for hit identification, hit-to-lead development, and lead optimization. In the present study, we use the human adenosine A2A receptor, which is involved in acute encephalopathy, Parkinson's disease, and cancer as a GPCR model. We were able to produce the protein at the milligram scale thanks to a baculovirus expression vector system optimized for production in SF9 insect cells. We used a 25-liter bioreactor to increase cell density up to 8 million cells per milliliter by controlled oxygen and air injection and the monitoring of dissolved oxygen and pH. Our A2AR is full length and wild type with a codon optimization for the sequence for expression in insect cells. The gene is fused to a self-cleavable signal sequence to enhance expression levels and protein targeting to the plasma membrane. We have also added two purification tags, a polyhistidine tag and a strep tag, which can be removed by a TEV protease cleavage site if needed. After protein expression and cell harvest, we isolated the cell membranes and screened for the best conditions for solubilization and stabilization. Here you can see by Western blot analysis how the protein behaves in the presence of detergents. For each condition, we analyze the total membrane proteins after 2 hours incubation and the resulting supernatant, which contains solubilized protein after ultracentrifugation, and the pellet, which contains insoluble material. As shown here, in the absence of detergent, A2AR remains in the insolubilized membrane pellets. We also performed a control with SDS, which extracts high amounts of membrane proteins, but is not suitable for purification because it is a denaturing agent. We then evaluated the solubilizing properties of the detergent DDM in combination with cholesterol hemisuccinate, CHS. DDM is a commercial detergent widely used for purification and structure determination of GPCRs, including A2AR. While this mild detergent was shown to preserve A2AR activity, we were only able to extract 45% of the protein from the membranes. Among our proprietary reagents, we evaluated the CALIX-10 detergent, which consists of a hydrophilic calixarine-based polar head and a 10-carbon aliphatic tail. Thanks to the Calix detergent, we were able to achieve complete solubilization of A2AR, as you can see here. We then evaluated the stability of the protein, solubilized with a mixture of DDM, CHS, and CALIX-10, since in this particular case, we wanted to perform further purification with DDM-CHS only. So we subjected the protein to a range of temperatures and quantified A2AR by Western blotting. We showed that CALX-10 highly stabilizes A2AR with a stability gain of 12 degrees over DDM-CHS alone. So using the identified condition, we solubilized the protein and purified it using tandem affinity purification, consisting in a first step of immobilized metal affinity chromatography, followed by purification using a streptactin XT resin. All purification steps were performed using an automated Acta Pure system, which ensures high reproducibility between our batches and results in over 95% purity, as shown here by stain-free detection of total proteins in the sample and Western blotting of A2AR. Once purified, we want to check the quality of our protein, and this starts with a cryostability assay. Here, we used nano-DSF with the Tycho device, which is a fast way to monitor the quality of the protein by measuring the fluorescence of tryptophan and tyrosine residues while subjecting the sample to a temperature ramp of 30 degrees per minute. Monitoring the brightness at 330 nanometer shows that there is no aggregation or loss of the protein after up to 3 Friso cycles in liquid nitrogen, as we don't see a decrease in brightness here. In addition, we monitored the ratio between 350 and 330 nanometer to measure the inflection temperature corresponding to the thermal unfolding of the protein. This temperature did not vary over the cycles, indicating that A2AR remains conformationally stable without the need for cryoprotective additives such as glycerol. Next, we confirm the functionality of the A2AR. To do this, we have two options for measuring ligand binding. So we have microscale thermophoresis, MST, and spectral shift using the monolith X, and radioactivity. The advantages of the monolith X are that the measurements are performed in solution in capillaries with low sample consumption, but it requires labeling of the targets with a fluorophore and does not give access to the binding kinetics. On the other hand, radioactivity is highly sensitive with extremely low background, but it is really time consuming and requires radio labeling, which is quite expensive. For MST, we labeled A2AR thanks to a fluorescent Tris-NTA probe and used two specific unlabeled ligands, the agonist CGS21680 and the inverse agonist ZM241385. As you can see from this graph, we were able to generate nicely fitting binding curves and determine a KD of 21 nanomolar for ZM and 30 and 312 nanomolar for CGS. For radioactivity, we used the unlabeled protein this time and the radio-labeled ligands. We determined close KD with a KD of 4 nanomolar and 193 nanomolar for the inverse agonist and agonist respectively. Both experiments showed that our purified A2AR binds its specific agonist and reverse agonist ligands, indicating that the native conformation of the protein is maintained. This experiment also highlights the fact that the relatively recent use of MST and spectral shift is an excellent alternative to the previous gold standard radio binding assay for GPCRs. Finally, we performed a thermal shift assay, this time on the purified protein, using DSF in capillaries as for MST. Again, we labeled H2AR with the red fluorescent probe and measured the thermal unfolding of the protein with a temperature ramp of 1 degree per minute to obtain a robust measurement of the melting temperature. By looking at the fluorescence of the probe, we were able to follow the unfolding of the protein and demonstrate an improved stability of H2AR upon binding of its specific agonist, with a gain stability of 9 degrees, and its reverse agonist with a gain of 5 degrees. These observations are tightly linked to binding events and conformational changes, proving once again that H2AR is fully functional. So at this stage, the protein was ready for heat discovery experiments, and we supplied the protein to Maud for larger scale analysis. I now give the floor to Maud.
Thanks, Alexi. Now that we have a high quality protein, we also need a good quality fragment library to identify novel protein binders. Our fragment probe set library is composed of more than 2,000 molecules. As you can see here on this middle panel, our library covers a large chemical space, including not only the chemical space of published fragments, here in yellow, but also new chemical entities. It shows high 3D and non-planar characters. Our library also obeys to a rule that is called the rule of three. As you can see on this histogram, the molecular weight of the fragments are below 300 Dalton. The number of hydrogen bonds donor are below 3, and the number of hydrogen bond acceptor are also mostly below 3. So let's now move to the screening cascade we follow. We first did a quick assay development and optimized the assay to check the protein stability and assess the robustness of the assay. We then perform the screening in single dose, and we confirm the hit by doing a dose response experiment that will allow to rank the compound by affinity. Then we use TSA as an orthogonal assay to identify stabilizers and to obtain more information on the mechanism of action of the fragments. We did a functional assay. Finally, a structural biology approach was done to complete the biophysical cascade. As you may know, MST spectral shift technologies require fluorescent labeling of the receptor. But before starting a screening, several controls are required to assess that the protein labeling does not affect its functionality. Here, we use CGS 21680 as the adenosine receptor agonist that Alexi already mentioned, as a reference compound to validate the assay development and perform the time course experiment. As you can see on this table, the signal is stable up to 5 hours with an excellent signal to noise ratio and a stable delta ratio. The affinity of the reference compound is shown on the graph here, and you can see that there is no significant change over the time. We also confirm an excellent interplate data reproducibility and actually validated the experimental condition. Now that the conditions were validated, we could start the affinity-based screen. A subset of 826 fragments were tested at the final concentration of 300 micromolar in duplicates. The compound dispensing was done by our compound management team by eco-dispensing. This workflow speeds up the screen and limits the compound consumption and the manual pipetting. Hits were defined as compounds inducing a variation of response higher than 4 times the robust standard deviation of the DMSO control, represented by the blue area in the 1D screen, the grayish. Hits are shown as blue cycle, and it is important to note that in spectral shift, hits can be positively or negatively affect the response, which correspond to a blue shift or red shift of the emission wavelengths. But here, as you can see, most of the hits are inducing blue shifts. Our positive control, the CGS 21680, was regularly monitored to assess the A2A adenosine receptor stability, and it is depicted here. We obtained a hit rate of 9.2%, and then selected 81 fragments for the dose response confirmation. So dose response titration were then performed on the 81 fragments in duplicates. You can see, for example, on these 4 graphs of dose response curve for the 4 best adenosine receptors binders. These fragments show affinities in the micromolar range, and more than 56% of it were confirmed with the GRC, and we were able to rank the fragments based on their KD value. Here you also have reported the ligand efficiency, LE, estimated per fragment. So as a reminder, the ligand efficiency value corresponds to a binding free energy per heavy atom. So a high ligand efficiency higher than 0.5 is considered to be an excellent starting point for a fragment-based drug discovery project. Then thermal shift assay, or TSA, was used as an orthogonal assay to assess the effect of the fragment on the A2A adenosine receptor thermal stability. So TSA is a simple technique where the fluorescence of the protein solution is measured in a temperature gradient. Here we use the Prometheus penta from Nanotemper to monitor the protein unfolding based on its intrinsic fluorescence. The first derivative of the melting curve, represented here, enables the determination of the melting temperature. As you can see on this chart, our reference control, the CGS 21680, stabilize the receptor, strongly stabilize the receptor by more than 7 degrees. So then we tested the selection of 21 fragments in triplicates at 300 micromolar final concentration. And you can see on this histogram that the A2A receptor binders are mostly stabilizer, but 4 fragments were identified as non-stabilizer and 1 was classified as destabilizer, which may reflect different binding modes or sites. And now I hand it over to Vanessa. She's going to tell you more about the functional assays and the structural studies that were performed.
Thank you, Maud. As additional experiment, the selected fragment hit with a KD under 300 micromolar were tested also in functional assays by measuring intracellular concentration of cyclic AMP. And what we observe with this experiment is those fragments show mainly antagonist behavior, and you can see all of the IC50 are reported only with the antagonist assay. As a follow-up, I would like to share with you the docking studies that was performed using the published X-ray adenosine receptor with the reference compound. And what we observe is that some fragment hits show purine-like scaffold as the antagonist reference compound and non-purine-like scaffold. So interestingly, non-purine scaffold is interacting with the same amino acids than the purine ring of the reference compound. So from the medchem perspective, it offers a novel vector to grow and can be used as one of the axes for fragment growing. Finally, to conclude this webinar, we have take-home messages for you that we would like to share regarding the role of biophysics in fragment-based drug discovery targeting membrane protein. So first, the protein quality and activity are crucial for fragment-based screening. And here we are working with excellent protein made by Eurofins CALIXAR. As protein QC, we are using nano-DSF and MST. All of those approaches suggest a protein that is native and functional. And regarding the screening, the Spectral Shift technology is the method of choice for membrane protein, and it's a rapid identification of fragment hits. I hope you enjoyed the webinar with us, focused on biophysics applied for membrane proteins. For any question, requests, or discussion regarding your project, feel free to reach us directly at this email address. You can you can take a look also on different posters online. Thank you, and happy to take any questions.
Thank you all for your excellent presentation. We now have the pleasure to introduce our questions and answer session. Don't forget you can still submit your questions using the questions panel in the menu on the right. So let's take a look at the first question. One for Alexis. Why did you decide to go with a detergent solubilization instead of retaining the natural lipid environment in a copolymer? Do you think the membrane protein lipid interaction is an important factor when looking for small molecule interactors? Philip likes very much the biophysical analysis, but how do we know if activity is impaired compared to the cell environment?
Yes, this is a good question because both systems are great to study membrane proteins, but here with copolymers we don't we don't achieve solubilization of all kind of membrane proteins, and in some cases they are not compatible with divalent cation, which can be a trouble if you need, for example, magnesium in your system. So we wanted to evaluate the system with detergents here. And actually, we performed also MST assays on membranes of insect cells producing A2AR, because in this case, we have very specific ligands, which is great. And we could see similar, we could measure a similar binding affinity of CGS and DM to the protein into the native membrane of insect cells. And we could compare that with the purified protein. So in this special case, we know that in our conditions, the protein is in the same conformation as in the membranes.
Thank you. And for Maud, what is the ligand concentration used for the DSF experiment? In the meantime, Vanessa, is the fragment library proprietary or is it available to the public? Can you provide the contents? Do you sell the library or do you have it as a collaboration project? Is it customized for GPCRs or can it also be used for soluble proteins?
Yeah, so it's Eurofins Discovery proprietary library. So we can share our library, the SDF file, but it's applied for our project here internally and for client. So it's a fragment library that was made and selected by our MedChem at Eurofins Discovery, and we are using for a large set of targets.
And for Maud, how did you label the protein with CGS21680 for spectral shift assay via streptobadin, barbietin, or some other kind of labeling?
The protein has a his tag, so we use the red tris NTA kit from Nanotemper to label it.
And what is the ligand concentration used for the DSF experiment?
For the DSF experiment, we use 300 micromolar for the fragments and 25 micromolar for the reference compound.
And also for Maud, in the spectral shift assay, can you confirm that it's just with the receptor and detergent, no other special additives?
No,
Could
there
you?
is
No.
no, there is no other additives in the buffer.
For Alexis, is expressing membrane proteins your expertise? If yes, in shake flasks or in bioreactors? And what about protein quality and yield?
Yes, it is, and we do both shake flask or bioreactors up to 25 liters in one bioreactor, and of course the yield will depend on the protein. So, but usually for a bioreactor we expect a milligram scale production of membrane protein.
And for Vanessa, have you published this work, and would you also be able to provide a reference?
So no, it's not planned for now to publish something. We make some posters and some webinar, and then we will share with you on our website. But for now, no, it's not planned to publish this data.
And for Maud, which dyes do you prefer for protein labeling?
So it depends if your protein has a tag. It's always good to use a dye that can label the tag instead of doing a random labeling that could affect the protein functionality. But both have their advantage. So yeah, when it's possible, we use the his tag labeling kit, but we can use also label using the Malimil kit that target the cysteine, or using the biotinylated kit. We have a large choice to perform the assay.
And for Vanessa, do you compare binding parameters obtained by spectral shift with the one coming from SPR?
Yeah, for this specific case study, we didn't have time to compare with all of the technologies that we have. For now, we have only data on spectral shift and some melting temperature. And for now, we didn't plan to test it on SPR, because here we saw that we are consuming a very small amount of protein, and we know that for GPCR it's quite challenging to have a large amount of protein, so it's the suitable technology for GPCR, for binding studies.
And also for Vanessa, do you routinely screen at 300 Is it not too high?
So that's often we have this question. I will say not.
as we are working with fragments with a very high solubility, so the stock concentration is about 30 millimolar. So I will say no, there is no aggregation. But we can also increase this concentration for screening. Here, the good thing with Spectral Shift and MST is if there is any aggregation, what we will observe it with the X scan. So we have a kind of internal QC to check this aggregation if there is aggregation for one of those fragments. But I'm not afraid to work with 300 micromolar. That will be not the same if we are working with in cell-based assay or biochemical assay, of course. But here, not.
Can any of your assays give clues to the location of the binding fragments on the membrane protein? And this is for anyone.
It could be. It could be, I mean, by MST Spectral Shift, we can perform competition assay with some reference compound. So if we know where the reference compound is binding, we can set up a competition assay with this compound. That's the case for GPCR, but for kinase, for example, we will use a ATP tracer to monitor this competition and to identify allosteric or orthosteric binding sites.
And we can also perform additional experiment using nano DSF TSA because we identify compound that stabilize the protein, and for if there are different binding sites, we can expect an additive effect if we mix the protein with two different compounds. So this is something that we will perform soon.
And for Alexis, can you express nanoluciferase and do Brett assay on membrane proteins?
Yeah, sure. You can perform luminescence transfer measure to membrane proteins without any problem.
Vanessa, could you comment on the spectral shift, red or the blue shift? Do you observe any correlation between protein ligand interactions and the direction of the wavelength shift?
For now, we didn't observe any correlation. If there is binding, there is a shift on the red or on the blue site. And when we perform a single point, so a screening at one concentration, we can have positive or negative signal ratio. So there is no sense and no correlation.
And how do you know your fragment binding site as you model them into the known ligand binding site?
Sorry, can you repeat?
Of course. How do you know your fragment binding site since you model them into the known ligand binding site?
So for the identification of, we do some modeling to see where the binding of fragment can be. It's only a docking study, but as the A2AR is already well described and there are a lot of crystal structure, some docking study can easily be performed and give some trend or some indication where are the fragments. But obviously, we need to do X-ray structure with fragment to make sure that it's binding where we think.
Thank you. And for Alexis, are the GPCRs always tagged on the N-terminus? Are C-terminal tags also used?
We also use the C-terminal tags, but you must be aware that in some cases for GPCR, you can have a cleavage of the C-terminal part, but both can be performed.
And did you already screen GPCRs with SPR?
For now, we have internal case study ongoing.
Thank you. And do you have a preferential host organism? What about Pichia pastoris? Have you ever used it for membrane proteins?
Well, for GPCR, we usually use SF9 cells because you have a high expression level of the protein, and it's closer to human than Pichia pastoris, but we can also express in Pichia pastoris. I would say that yeast are more difficult to handle because they are tougher to break, and we usually prefer to use insect cells. But both system has been used for A2AR and both works.
And why did you select A2A? Are you taking these sound fragments any further in the drug discovery process?
I would say that A2AR, we know very well this protein at first, and also we performed all our first assays with MST here, Spectral Shift, with this specific protein, which has great specific ligands, and this is a great tool for it finding because you have great controls actually of the protein.
And would you be able to potentially identify allosteric modulators using the techniques discussed?
Yes, actually with MST you can have two different events. You can see different events, binding events. So if you see that if you see that you have a sigmoidal curve with two different events, you can observe these allosteric events.
And have you tried this workflow with different WT GPCRs?
Sorry, can you repeat the question?
Of course. Have you tried this workflow with different WT GPCRs?
Wild type.
Well, the whole workflow with here we work with GPCRs and biophysics, MST, and all that stuff a lot. But so far, for the whole hit finding assay, we did not yet try another GPCR, but we are open to it.
Thanks, Alexis. And also for you, are there any specific requirements for the recombinant proteins to be compatible with the spectral shift approach?
Not really. You only need to label, either label the protein with a fluorescent probe or have fluorescent ligands. This is also a possibility. And to label the protein, you have many options, as Maud said earlier. So you can use the purification tags, yeast tags, trep tag. You can use also biotin, or you can just label the protein directly with NHS or maleimide. So you don't really need any requirement.
For protein that bind DNA, we can use labeled DNA and follow the binding using the labeled DNA instead of following the protein.
Thank you. And for you, Maud, can we use up your fragment library with other biophysical technologies such as SPR, for example?
Sure, the library is compatible with all the biophysical technique that we have and that exist. The fragment library is so has been we monitor the purity and the solubility of the fragment, so they can be used for SPR or for TSA. Sure.
And which mentioned technique has the highest speed and throughput in screening?
I will say Spectral Shift. For the plate and the fast readout. So typically here the spectral shift can be used for fragment library, but also for large set of compound library.
Fantastic, thank you. Please join me in thanking Vanessa, Alexis and Maud. As you leave the webinar today, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.
Goodbye.
Thank you very much. Goodbye.
Goodbye.